Team Bio, sorry I couldn't be in class today to go over the lab with you. So I'm going to just record a quick screencast going over the explanation for what we saw in every part of the lab. Okay, so the first round of the lab having you adding water to fresh liver, and most of you predicted correctly that there would be no bubbles. And the reason for this is that there is no substrate in this reaction. Oh, Roomba's getting active. Nope. Without the presence of H2O2, the reaction cannot take place. Because remember, catalase catalyzes the decomposition of H2O2 to water and oxygen. So without the presence of hydrogen peroxide, there will be no gas, so no bubbles. Okay, second round. We added liver to hydrogen peroxide and we did it 2 times. And we saw bubbles in round 1 and bubbles in round 2. And the reason for this is because, remember, catalase is a catalyst. And a catalyst has there are 2 features of being a catalyst. One is it speeds up the reaction. And 2, it is not consumed in the reaction. So when we added H2O2, we got bubbles, and then we kind of let that reaction go all the way through. So all the H2O2 was decomposed, and then we wiped off the liver and scooted away the bubbles, and we added more H2O2. And so catalase was still present, and now it has a fresh round of substrate, so it can continue to catalyze that reaction. Some of you might have noticed that the bubbles round 2 may be slightly reduced. And you also might have noticed that the liver started to turn brown. The reason for this is this reaction is an exothermic reaction. And it's releasing so much heat. You're putting on so much substrate, much more hydrogen peroxide than would ever be kind of naturally occurring in the body at one time. So this reaction, round 1, created so much heat that it burned, or it burned some tissue. And it might have denatured some enzymes. Not all of them, because we're still getting another reaction in round 2, but a few of them. And the less hydrogen peroxide you add at a time, the less likely this is to happen. That will produce so much heat that will basically burn the liver. Okay, but in the absence of this, if this is not a strongly exothermic reaction, or we just add tiny little bits of hydrogen peroxide at a time, we should have no damage to the catalase, and we should be able to keep adding more and keep getting more and more bubbles. But if we add too much, we run the risk of denaturing the enzyme, breaking it apart because of the heat released. Okay, in this third iteration of the experiment, I had you cut the liver up. And a lot of you predicted that you would see lots of bubbles. And they would be big, indicating a large reaction. And the reason for this is by cutting up the liver, you're actually increasing the surface area. And the number of places where H2O2 can interact with catalase, so the number of places where hydrogen peroxide is now touching molecules of catalase is more. So basically, the hydrogen peroxide can be decomposed by the catalase faster because there's more enzyme at the surface. So this reaction happens really quickly, and you see lots of giant bubbles. Okay, when you added hydrogen peroxide to the boiled liver, you saw no bubbles, or very, very small, very limited reaction, very small bubbles. Hopefully you all understand after watching your screencast from last night that boiling the liver denatures the protein. And so we've taken catalase, which is this very complicated three-dimensional structure, and we've broken all of those hydrogen bonds that are holding it together. We've turned it back into a polypeptide, that long linear chain of amino acids, which can't perform its function of decomposing hydrogen peroxide. So boiling liver is going to create no or very small reaction. I think for some of the pieces of liver, I didn't boil them quite enough. They might have been still a little pink inside, which is where maybe very small isolated places didn't get quite hot enough to fully denature the protein. So some of you saw very small bubbles, but many of you saw no bubbles at all. Okay, same thing with the liver covered in hydrochloric acid. When we saw no bubbles or very limited bubbles, and the reason for this is that hydrochloric acid, just like the heat, is going to break hydrogen bonds and denature the protein. So that shape, which is so important, the three-dimensional structure, falls apart and it can no longer do its job, and therefore we see no reaction. Okay, this last one, hydrogen peroxide added to the frozen liver, was a pretty interesting one. I think most of you noticed that when you added hydrogen peroxide right away, the first thing you saw was like a fizz. So tiny, tiny bubbles. Which indicates that the reaction was happening, but it was happening very slowly, which makes sense because when the liver is frozen, the enzyme can't flex as easily. So it can't undergo its conformational change that it needs to do in order to actually catalyze this reaction, because the bonds are literally frozen in place. So the reaction happens very, very, very slowly compared to these other reactions at room temperature. However, this is an exothermic reaction. So as the liver as the catalase in the liver starts to decompose the hydrogen peroxide, this creates heat. Or I guess it releases heat. Because energy can neither be created nor destroyed. Okay, so this releases heat. So then the enzyme starts to warm up. So then the reaction starts to go faster. And then we ultimately see large bubbles. So it goes from very distinct, teeny tiny little fizz to huge bubbles over the course of the enzyme heating up. Okay, that's it. Hopefully this lab really impressed upon you the importance of enzyme shape and its function and all of the different factors that make it possible for the enzymes to retain their shape or denature their structure. And hopefully you see the importance of, in living organisms, the fact that we need to maintain homeostasis. So having a consistent internal temperature and having a consistent internal pH is super important to maintaining proper enzyme function in our body. Okay, that's all. Hopefully I'll be back in school tomorrow to answer any lingering questions you have about this experiment. Talk to you guys later. Bye.